And thank you. Actually, Robert Reed suggested me to come over here, so that's how I'm here. Can you hear me?
Just gotta put it down so it's
okay. I'm going to start with a little bit of introduction. Okay, so this is my yard in somewhere around late June and July. So you can see this is not a lawn, but contains lots of native plants. So it's more than 200 species of plants are there in this yard. So if you have such biodiversity, then you have a lot more other insects and other creatures which depend on the plants. So I just wanted to tell you what naturalist is. So naturalist is one who makes observation of relationship between organisms and their environment. But natural things could be anything, like even volcano or like eclipses and things like that. So this is what made me interesting, and I thought like I should share this with you. Like curiosity leads to discovery, and discovery leads to knowledge. Knowledge leads to insight, and insight leads to understanding. This was quoted by Stephen Shepard, who is the host of Natural Curiosity Project for Apple Podcast. And curiosity is the most important thing if you want to learn anything. So first you can become naturalist, and then you can continue your interest furthermore and become a scientist. That's how I have become. Okay, can you show the clip? This is a clip which was recently published in Science magazine just a few days ago. So this is a caterpillar in the front. Caterpillar in the front is the one which was originally on this plant. So scientists put another caterpillar here, and then put a sound recorder here, and watch what happens. So this is the sound you are hearing of the caterpillar making on the leaf. But wait and watch what happens afterwards. So generally on this leaf there are only one caterpillar. But if somebody else comes, they have to defend their territory, and that's how they do this dance. And he hasn't gone very far. He's just down there. And so this Science magazine just had information saying that it didn't tell me what happened after that. So I was keen on knowing whether after that what happened to that caterpillar, whether it came back and chased away the other caterpillar. So that is the curiosity. You keep looking for more things as you see something. So observation is important. So these days, natural history has become quite an interesting thing. Almost everybody is interested in natural history, but they use different techniques than what I used to use. I used to mostly use books and things to identify. But now, cameras have changed the whole world. Like, for example, you watch that video. So video can show a lot more about behavior and biology. People started with point-and-shoot cameras, which can do a very good job with photographing these things. And then professional people use more macro lenses and things like that. But even cell phones like these can be used to record all these observations. And very often people who start with this, they don't know what they're looking at. So there are a variety of websites where you can upload, and those websites can help you. So for example, most of these people do on, where I didn't put, iNaturalist. iNaturalist is one of the important website where you can add your photos, and that will tell you. But you shouldn't stop just at identification, but you should go ahead and look at more observations and things like that. Here you can also see CUIC, means Cornell University Insect Collection. That also can help you with identification of your images. Mostly these are done by computer vision models and AI models. So iNaturalist has become a lot more important these days because lots of pictures have been added to that. It's not only pictures, but sounds and videos also can be added to some of the websites. So because there are so many informations available, citizen science is becoming a lot more important because nature contains so many things. All scientists put together can't study all those things. So it is it's the citizen scientists who need to learn more and add more information to these kind of observations. So, for example, I'm talking to you about my house. I have been recording last 6 years on iNaturalist, whatever insects come to my place. And all plants I have grown, I have made a record of all these things. So based on this, I have 5,047 observations, of which verifiable species are 1,111, and research grade are 724 species. So you can see just my house is, my yard is just about less than half acre, but you can get so many things if you have your native plants native things in that area. So that was the website. So this film we are talking about, so I'll talk about a little bit of the area where this film was recorded. So this is India. India has very diverse habitats, including desert in this part of the country, while these mountainous regions have evergreen forests. This is tropical forest, and these are temperate subtropical forest. And so here you see, this is a close-up of eastern part of India, where biodiversity is very high. It is supposed to be a biodiversity hotspot because it has influence of Indian subcontinent's flora and fauna, as well as from Tibetan plateaus and Malaysian region. This is called Malaysian region. So this place is very interesting, and the place you're going to see movie today is from this area. So I actually was, in one of these parts of the country called Sikkim, which is just like 80 kilometers wide and 100 kilometers long, and land goes from 200 meters to 8,000 meters, and it is very diverse area with 650 species I had done, but now it is more than that. Based on this, I did some study for this, and I wrote a book about this area called Butterflies of Sikkim, like which Molly referred to. But I also wrote a book of damselfly and dragonflies of this area. Here we have something like 120 species almost, and this is a revision I'm going to do. Should be available next couple of months or so. So, but mostly I did daytime work with the butterflies, but recently I went to, again, that same area where I did some mothing, and it was so amazing. Like when I was in Sikkim, somebody knew I was collecting butterflies. So one of the shopkeeper came one day in the morning with a big container full of moths. He said, like, these are the moths which came to my light. So there were some live and some dead moths, but it was full of that container. So some of these things you will see in the movie. This is called tropical swallowtail, feeds on macaranga plants. And diversity is so amazing. Just within a night, I had something more than 150 species. So it's very interesting, but things are changing because lots of deforestation and development is taking place. I'll let now Jason to talk about moths to you. Oops.
All right, can everybody hear me back there? Great, okay. So Meena gave us a nice introduction to what everyday people can do to contribute to the science and gave you a nice introduction to the area. All right, so I'm going to give you an introduction to what a moth actually is. So I'm going to cover these in a little more detail in the next few slides, but basically moths are a group of insects that belong to the order Lepidoptera. Lepidoptera also includes butterflies, and we'll go over that a bit later. Lepidoptera means scaly wings, so they're covered in deciduous scales. They often have a proboscis for their mouth parts and a four-parted life cycle, which we'll look at. But I just want to sort of start with touching on the diversity. 180,000 described species. So that's species known to science. Of those, about 20,000 are butterflies. So that leaves 160,000 moths. There's probably almost as much more that are undescribed, especially in tropical countries. But even locally here in Ithaca, you can get species that are new to science. All right, let's talk about scales. So I said Lepidoptera means scaly winged. Indeed, moths are covered in scales. Let's see. Oh, there we go. So this is a close-up of the wings of a moth. And what you'll see, it's got all these overlapping scales, which creates the color and pattern on the insect. Now, it's not just covering the wings, it's covering the body. So here's the head of a lovely little leaf roller moth. There's some of its mouth parts there, antennae up here, there's a wing, and there's a leg. The whole insect is basically covered in scales. And these scales can rub off, they're deciduous. And so if you took all the scales off of a moth or a butterfly, what you'd end up with is a shiny brown body, shiny legs, shiny antennae, and clear wings. Basically, the whole thing is covered in scales. Now, you might have heard the myth that, oh, if you take the powder off of a butterfly or moth, they can't fly anymore. Not true. They still can fly, they're just ugly. All right, mouth parts, proboscis. A lot of moths have a proboscis. Here it is right here. So here's a lovely little grass moth, and its proboscis is uncoiled and drinking some nectar. Here's a sphinx moth, where it's coiled up underneath the head like that. So basically it's hydrostatically controlled, and there's sometimes musculature inside that can control it, and they use that basically as a straw. And the characters I'm talking about all refer to the adult part of a moth, but butterflies and moths have a four-parted life cycle. So here is a local species. This is the forest tent caterpillar. Up on the top left, we have a mass of eggs. And this you can find right now. If you go outside, look for some cherry branches, you will find masses of forest tent caterpillar eggs. This is probably about 100 or 200 eggs. Those eggs hatch into a caterpillar. And there it is right there, lovely little caterpillar. By the way, in this species, the way to recognize it is it looks like it's got little penguins down its back. And so that's the forest tent caterpillar. Now the caterpillar is the eating stage of the animal. So basically they eat and grow and eat and grow and eat and grow, and they eat a huge amount of food and eventually store a lot of that as fat. Then they go into the pupal stage. So inside this cocoon, so the cocoon is a silken structure, there is a pupa, and that pupa is sort of intermediate between a caterpillar and an adult, and so all kinds of old body parts are being broken down, new body parts are being developed, and then eventually the adult emerges. And here's an adult forest tent caterpillar moth. And basically, the adult is often the shortest part of the life cycle because basically all they need to do is mate, lay eggs, and they're done. That's all there is to it. Of course, there's exceptions to that. But yeah, four-part life cycle. Egg, larva, pupa, adult. Okay, now we talked about diversity, 180,000 species known globally. Well, what's it like in New York? Well, we don't really know. We're still working on it. The last true census of the butterflies and moths of New York was done over 100 years ago. A colleague of mine, Hugh McGinnis, and I are currently surveying to update it. We've added about 1,000 species to the list, and so we're currently at 3,200 species. If I had to guess, I suspect there's probably close to 4,000 species total, and various parts of the state are more diverse. Now, it's not that there's no moths down in the lower left corner of the state in Chautauqua County. It's just very few people have gone there surveying for moths. But definitely Long Island is much more diverse than the rest of the state. All right, so how do we go and survey for moths? Well, this film will explore one of the ways, and that is using a light. So there's various different types of lights you can use. On the left, this is a LED light. This is some of the more modern types of lights you can get. And this is one I set up in Chiapas in southern Mexico. You can see it's quite busy, so it had a lot of stuff at it. And you set it up against a sheet. The sheet helps sort of project it more and also gives a place for the moths to land where you can actually see them. In the middle, that's my backyard. A little different than Meena's, not as much cool wildflowers, but that's my setup with the mercury vapor light and again that sheet. In the day, it's not that busy, but also not as busy as the tropics. And then you can also set up various traps. So on the right, this is a bucket with a funnel in it, and there's a light there. Moths come in, go into the bucket, and then you can look at them in the morning. That's a shot I took in Idaho on the Salmon River. All right, and the last thing I'm going to end off on is if you're impressed by this movie and you want to do mothing of your own, you can just turn on your porch light. It won't do as well as a true mothing light, but that's a start. Or I do public moth nights. I've got 2 scheduled this year. The first one is on the 31st of May, and a rain date of the 1st of June with the Finger Lakes Land Trust. It's probably going to be at Bell Station or Cayuga Shores Wildlife Management Area. That's TBD. I should know in the next few days. The other one I'm going to do is up in Seneca Falls at the Seneca Meadows Wetland Preserve on the 27th of June. And at these mothing nights, you basically you bring a lawn chair, a nice comfy beverage, and a light, and I put a light up, and we talk about what moths come in, and I tell you all kinds of tales of the cool things that they do. Now, just to warn you, the sheet will not look as crazy as it probably does in this movie, but it will look closer, hopefully, to something like this. This is my sheet in my backyard, and it should be comparable to that. All right, with that, I will pass the torch on.
You should definitely go black lighting if you've never done it before. It's one of the best things you could do in life. So I'm just going to give maybe a 5-minute tour of the research I do in my lab here on campus. I've spent my whole career studying a very small group of moths that are called butterflies. They're like day-flying moths. You may or may not appreciate, well, I guess Jason just gave you the rundown. These things are covered with scales, right? This is a close-up image of a butterfly wing. Here's the laser. And their color patterns are basically mosaics of thousands and thousands of scale cells. Each cell is a different color. And we have these patterns of spots and stripes, and each cell along its developmental pathway decides what pigment it's going to express. So we have these beautiful color mosaics. So what I study is where color patterns came from and how they develop. Okay, so if you look at all the butterflies you see in nature or look in a drawer of butterflies like the one in the back, they all look really different, all these different colors and patterns, spots and stripes, different pigments. But it was realized going back to the 1920s that all of these different wing patterns can be connected to each other through what we call these days the nymphalid ground plan, or the butterfly color pattern ground plan. And this is an image from a Russian entomologist named Schwanwich up here that shows this ground plan here. So you can get all of these different color patterns just by moving these spots or stripes up and down the wing or changing the size or color of these individual elements. You can get butterflies like this that have gigantic eye spots, or some butterflies that look like leaves, right? There's no eye spots, and these stripes that become small and kind of dark red. So you get all of this diversity we see in nature with this ground plan. So in my lab, we're really interested in the genetic basis of this ground plan. Where did it come from? Where does the color red come from in these insects? And butterflies are a pretty good system for I think, addressing some of these really fundamental questions in biology, ecology, and evolution. So just briefly, we get funding and interest for the kind of work we do for a lot of reasons, right? Butterflies are a nice model for looking at biological pattern formations. Their wings are a nice flat sheet of cells, a two-dimensional surface, and you get these emergent patterns coming out, like these concentric circles and these symmetry systems of stripes, things like that. So we're interested in how cells talk to each other to make these patterns. A lot of labs study population genetics. We're interested in the biology of seasonality and how different color patterns can change depending on the time of year and how the butterflies know what time of year it is. We're interested in biodiversity, of course, and the movie's going to touch on that. And also just the origin of newness, right? So in some butterflies, you see these totally new traits or colors appearing. So we're interested in how new things evolve in species. So I'm going to show you a couple slides of pictures of the kinds of stuff my lab does to ask some of these questions. I won't go into too much detail, but if you open the side of a caterpillar, just make a little incision on the side and peel it open, there's actually a little tiny wing inside the caterpillar, right? And when that caterpillar pupates, the wing gets really big and it starts developing its colors and stuff. But these are wings that I removed from caterpillars. So I literally just cut open the side and pop these out. And they're just like little tiny wings, right? So this would be the wing hinge where it connects to the body. And this is the edge of the wing here and the wing veins, right? And even at this point of development, the genes are starting to talk to each other, saying, okay, there's going to be a spot here or a stripe here. And I'll show you some pictures of gene expression in just a second that'll demonstrate that. And then this is a chrysalis of a heliconius butterfly that we've done a lot of work on. And in these images here, I've just peeled back the cuticle from the side of the pupa, and you can see the wing developing its color over time, almost like a paint by numbers, right? So they start out really thin and clear. You have this nice sheet of epithelial cells, and then the scale cells start to inflate like little balloons almost, and they start developing their pigments, but in a very specific and stereotyped order where you get the red and the yellow ones first, and then you get the black pigments appearing in a wave coming out from the middle of the wing. But even at this point, all the cells know what they're going to become. All right, so here's just a nice image of a butterfly eye spot. So how are these stripes and colors made? It all has to do with the genes, okay? So here is a caterpillar wing, just like I showed you before, but stained in purple is the expression of a gene. This particular gene is called Wnt, if you care about genes. So it encodes a protein that's expressed from the cell, and then it seeps into other cells around it, telling them to take on a new identity. This is a common buckeye butterfly, so one that kind of occurs around here. It's more common to the south. This is a caterpillar wing from this butterfly here, and you can see that this gene expressed in purple here, it's a purple dye, is expressed in the stripe pattern down the middle. And then here we have the stripes at the edge. And then this little stripe here, notated B, is that stripe right there. Right? And here's the hind wing. And we could use methods like CRISPR genome editing now to go in and we can delete the gene or we can erase it to ask what its function and color pattern development is. And so this is a mutant that we created in the lab, a transgenic butterfly, where we deactivated this gene. And you can see that those stripe patterns are lost. So focus on this big black central stripe pattern that goes all the way down the middle here. It's totally lost if we take out that gene. These edge patterns here, like one of these edge stripes is gone, like that thick brown stripe right there. And then that little one at the base right there is missing. And on the hind wing, you can see this like big kind of beige stripe right here. That's missing. All right, so these butterfly wing patterns are highly modular. There's individual genes that control like the eye spots, the different stripes, and all that kind of stuff. And we could remove them, and we could add them, and we can change the color pattern, almost like a Lego set. And then we have a butterfly like this. This is a painted lady. It's kind of a psychedelic mess. It's hard to make sense of here. But you see this stripe pattern again down the middle, like where this would be. And this is kind of fun. When we knock out the gene here, you can see that whole like central stripe pattern goes away, right? So there's the mutant that we made, and then there's the wild type, right? So that's a single gene that does that, making the stripe down the middle. Okay. So that's an example of a pattern formation gene. Here's a pigmentation gene. This is a gene here called optis. It's expressed here. We have a dye. green dye here showing you expressed in Heliconius athis that makes these nice diamonds on the hind wing. and this one is correlated with red. So here, let's knock it out in this red butterfly. This is a Gulf fridleri, and when we knock out this like paintbrush red gene, you get a totally black butterfly, right? These do not exist in nature. This is the most heavy metal butterfly ever, right? A completely black butterfly. So this gene optics, it makes the color red, right? So upstream of it, you have genes like Wnt that make stripes, and then they activate these paintbrush genes that will make yellow or red or black or whatever. And this is a switch gene that switches between black and red colors. So here's one of those painted ladies. Here we made a bilateral mutant here. So it's a mutant on one side. You have optics turned off. And it looks like a photocopy, right? It's like black and white. There's this one gene that makes all the color, right? So this is mostly like a wild type side, and here's the knockout side. So it's incredible. So here's the normal wild type one, and that's the mutant. So just all the colors missing. There's just one color gene. We were very surprised to find. Here's just the last little example. We knocked out optics in a Gulf fridleri, right? So you'll expect if we knock it out, you know, the orange and red pigments will probably go away, and they do, but then it turns bright blue, right? So this is like an iridescent color made by scale structure. So this is a super interesting gene to us, just one of several examples of these interesting color pattern formation genes. It controls not only pigmentation, but also structural color as well. So it almost turns into like a little morpho butterfly. It's a big surprise. So yeah, there's one master gene that controls all colors. And so we were pretty excited to find that. And that was kind of cool to see, because if you look throughout all the butterfly and also some moth diversity as well, you see this kind of archetype show up over and over again in moths, all these totally unrelated, butterflies. I don't know, are there many moths that look like this? Like black and blue, there's a few. But these are all totally unrelated butterfly groups from different families, and you see this black and blue archetype show up over and over and over again throughout evolutionary history. And now we know there's only one gene that's required to do that. It just turns off all the color and turns on blue. So hopefully that's a kind of an entertaining little vignette of the kind of stuff we're doing. You could dig a little bit deeper into like any of these moth or butterfly groups and find all kinds of interesting surprises and things that we can learn about how biology works. So with that, I guess we could turn the lights down and head to Arunachal Pradesh, and you can see some cool black lighting. One of the things I really like about this movie, so it looks cool, right, just to see all the moths come in the black light, but also just listen. Just listen. So the directors of this movie set up microphones on the sheets, and you could hear like the rain, and you could hear the moths like coming in and hitting the sheet, like all in these like really long kind of meditative sequences. It's really, really cool. So yeah, enjoy the movie with all of your senses.
And just before we do, please join me in warmly thanking Bob, Meena, and Jason for sharing these insights with us.